Hello, my name is Marie, and my lesson today is on skeletal muscle fiber types. I want you to recall what you know about cellular respiration and the basics of glycolysis and the electron transport chain and the making of ATP oxidatively. There are really two routes that a cell can take. So ultimately, glucose is our energy source, and glucose will be broken down into ATP either glycolytically if there is a lower level of oxygen, which will follow this route here, and NAD is replenished, making very little ATP, as opposed to the oxidative way. When oxygen is present, cells will use mitochondria. There's an electron transport chain. NAD and FAD are electron carriers that help to produce about 32 molecules of ATP per molecule of glucose. And it's easy to understand the fast and slow twitch muscle fiber types when you understand cellular respiration. Most people would agree there are three types of fibers. Some may say two, fast twitch and slow twitch. But we will talk about three types of fibers in this lesson. And the classification of these fibers is based on how they synthesize ATP and hydrolyze ATP. So it's the enzymatic machinery that they're using for making ATP. And that allows the muscle fiber to either contract slowly or rapidly. ATP is constantly being made in our bodies in a cycle that never ends throughout life. And it's the hydrolysis of ATP knocking off a high energy phosphate bond that allows for energy to be transformed into the energy for cellular work. And in the opposite way, when phosphate is bound to an ADP molecule, we make ATP. There are three major types of fibers: slow oxidative, fast oxidative, and fast glycolytic. And the slower fibers have many mitochondria in them, and therefore many capillaries supplying blood, which gives them a reddish color, and their contraction is rather slow. They have a high resistance to fatigue, and they can work for a longer amount of time as opposed to the fast oxidative and even fast glycolytic fibers. Fast twitch fibers can be either oxidative or glycolytic depending on how they're making ATP. And these are fibers, the fast twitch ones use mainly just glucose as opposed to fatty acid and glucose, which are the slow oxidative fibers. And the fast glycolytic fibers have a high myosin ATP ACE activity. Their contractions are fast. They have a low resistance to fatigue, which means those muscles will burn up that energy and then not be able to work much after that. They have a low oxidative phosphorylation capacity and very few mitochondria. Fast oxidative fibers, the type 2A fibers, are somewhat in between, but they are using oxygen at a higher rate than the glycolytic ones. On a practical level, coaches, trainers, elite athletes, athletes really of any kind, are interested in the muscle fiber types because humans and mammals either have a higher percentage of fast twitch fibers and slow twitch fibers, and that's a genetic endowment that people have. So when we're looking at athletes, there's really a continuum that we have. Some people have more slow twitch fibers, some people have more fast twitch fibers. We all have some of each, but there is this continuum here. And there are coaches that would probably say there are even more types of fibers. There are type 1, type 1C, 2C, 2AC, type 4X, 2X type fibers. And it's on this continuum here. And here are some interesting photos of some long distance marathon runners, which you could imagine most likely have a higher percentage of slow twitch fibers. This is Desiree Linden and Yuki Kawachi, who ran the Boston Marathon in 2018 in just over 2 hours. So if you can imagine running for that long at a pretty steady pace, it's pretty amazing. Whereas Usain Bolt, his average speed was 23.35 miles per hour at his fastest. So, for most of us, if we want to lose weight, we need to actually exert our slow twitch fibers for a longer period of time, and um, and that's cardio exercise that's necessary to actually burn fat because fatty acid metabolism is taking place once you do that, once you're working those muscles for a while. There are three basic pathways for these fast and slow twitch muscle fibers, and our bodies will use creatine first, and um, and it is really the first molecule that's used to supply ATP. It has high energy phosphates. It's a reversible reaction. However, it runs out quickly. If oxygen is present and plenty of oxygen, then our bodies will do oxidative phosphorylation, producing lots of ATP per molecule of glucose. And then, if the supply of oxygen cannot keep up with the demand for oxygen, our bodies will do glycolysis. And here is a micrograph showing various kinds of muscle fibers, and the slow oxidative ones, and even the fast oxidative ones to a degree, show up fairly darker because of that myoglobin and higher concentration of mitochondria. In fact, even meat that we eat, like in chickens, the dark and light meat is a reflection of how much mitochondria is in those muscles. The fast glycolytic show up as being very light in color. This is a graph just showing the time it takes for muscles to contract, and uh, some muscles will get fatigued. They all reach the same force, however, they do so in differing amounts of time. So throughout our bodies, we have various kinds of muscle fibers, fast twitch fibers in small muscles like our eye muscles, and deeper muscles that are able to work for a long amount of time in our legs are slow twitch fibers. This is an interesting study where scientists at the University of South Dakota were trying to figure out whether different kinds of muscle fibers release varying amounts of myokines. And they were looking specifically at interleukin 6, which is a well studied myokine known to regulate muscle metabolism. And we usually think of muscles as being really their purpose is for locomotion and posture. However, muscles can also be endocrine, um, glands, even. So, myokines can be produced in an autocrine fashion, a paracrine, and even an endocrine if it gets into the blood, and actually has huge benefits for metabolic disorders. And in this study, they used male and female mice, and they wanted to establish, their purpose was to establish whether there was an ex vivo approach that they could do as opposed to in vitro and in vivo. And if they isolated two kinds of muscle, could they show with a stimulant whether it was producing interleukin 6? And interleukin 6 is this myokine that can be anti inflammatory and pro inflammatory. And what they did was they isolated the muscle, and they placed it in a Krebs solution with and without oxygen. So part of their experiment was oxygenating it, but also then varying the amount of temperature. And the results show in a Western blot here that they had a soleus and an EDL muscle. So The soleus muscle is a calf leg muscle behind the gastrocnemius. It is known to be a slow oxidative muscle, and the EDL muscle is an extender digitorum longus muscle near the tibia, and that's known to be a fast glycolytic muscle. So they first validated whether the ex vivo approach was feasible. They measured the IL-6, and then their results showed that the soleus muscle was definitely higher in the amounts of IL-6. The C here is the control. This is a stimulant, and they were using various kinds of stimulants, including nicotine. This is the stimulant here, and here is the control and the stimulant with varying amounts of whether it had oxygen or not, and two different temperatures that they used here. The ELISA assay also showed that with the stimulant, it was definitely producing the IL-6. And you can see here that the soleus muscle was showing greater concentrations. This protein was showing up in the soleus muscle as opposed to the EDL. And this immunofluorescent image here showed that the staining showed a stronger IL-6 signaling intensity in the oxidative muscle fibers. So the authors concluded that the oxidative muscle fibers were definitely producing more IL-6. However, they say for further study that there could be other stimulants that might work on the EDL muscle. These are my references, and thank you.